Good morning. Let's get started. So we're very pleased to have you here today. Yue?
Yes.
Hi. Sorry, you're frozen on my screen, so I just wanted to make sure everything's okay.
Sorry, I think me. Can you see me now?
Yes, much better. Okay, so we're going to get started. Good morning, and we're really excited to have you here with us today. We're doing two special presentations on the Tapestri platform to accommodate all of our customers in Asia and in Europe. To begin, we have Dr. Yue Wang, who is a field application scientist with Mission Bio. And today she'll be talking about an introduction to the Mission Bio Tapestri platform and its multi-omic capability to detect SNV, CNV, and protein using single-cell sequencing.
Thank you, Kelly.
Just as a reminder, we're currently recording the session and streaming live on YouTube, and we love to answer your questions in real time. And so if you have any questions, please feel free to enter them into the Zoom chat or Q&A box, and we will answer them during the seminar. All right, so with that, I'll stop sharing, and Yue, take it away.
Let me share my screen. Can you see my screen?
Yes, it looks great.
Okay, great. Thank you. Thank you very much, Kelly, and good morning, everyone from APAC region, and good evening if you're from the US region. So like Kelly mentioned, today I would like to talk about how to use Tapestri platform to perform multi-omics analysis of SNV, CNV, and protein sequencing from thousands of single cell. Just a brief overview of the agenda today. First, I would like to give a brief history of our company and why do we want to use single cell. Next, I would like to talk about two custom publications and share the data with you, focusing on therapeutic resistance and MRD studies. Finally, and most importantly, I would like to share our current progression on single cell omics, multi-omics study. Just a little bit back history about our company. Back in 2017, we launched the first single cell genomics platform called Tapestri. We often get the question that how we're different from other single cell company in the industry. And the answer is that we mainly focus on single cell DNA. And we were the industry first single cell targeted DNA sequencing platform with several catalog panels being offered targeting both heme and solid tumor space. In 2018, we launched Tapestri Designer, which really unlocked the potential for custom panel designs for high impact application, including CRISPR screening and mouse studies. And major interest using our platform lies in translational and clinical research. And more recently, in 2019, we are hard at work to launch the industry first DNA-focused multi-omics platform. In here, we combine the information of both DNA and protein at the same single cell level so that allow customer to analyze genotype and phenotype at the same time. This really launched us into preclinical and biomarker space. With single cell DNA at our core, we're really excited to drive the Tapestri platform into multi-omics space. So why single cell DNA and what's the benefit, especially in cancer research? The current popular method to understand cancer genetics is bulk sequencing. However, this approach does have several limitations. First, the conventional population average approach failed to resolve potential minor population, but those rare cells or small clones sometimes could be very important and informative for clinicians to make relevant intervention. And second, it fails to resolve the subpopulation makeup in a highly heterogeneous cancer sample, both blood and solid tumor. And in bulk sequencing, again, population averaging may just obscure the important biological information, and usually very true for single cell genomics analysis of a diverse heterogeneous tumors. In a recent review that just came out of Nick Navin's group at MD Anderson, he reviewed for the first time how single-cell genomic sequencing can be potentially applied to the clinics. And he talked about all these applications from DNA to RNA to epigenetics. But today I really want to focus on how single-cell DNA can be especially used in your clinical research. On the right graph, it has this nice summary of the 4 categories that you can use single-cell DNA sequencing to generate. At first, single-cell DNA sequencing technology can help you to resolve different clones from a mixed population. And based on their mutation acquisition order, further, you could even construct phylogenetic trees to better understand tumor evolution. And if you're studying with longitudinal samples or different spatial samples, you could also construct fish plots to better understand the clonal size change at different time point or different space in same patient. And lastly, also very importantly, using single-cell DNA, you're able to resolve the mutational co-occurrence pattern and even resolve the zygosity of each mutation. Again, just want to show a list of specs that showcase Tapestri platform's capability. As the industry's only platform that analyzes single-cell DNA and protein at the same level, our platform allows assaying up to hundreds of targeted loci for DNA analysis. And we also allow analysis of CNVs at both gene and chromosome level. And our high throughput allow you to analyze up to 10,000 cells at the same time. And moreover, the high sensitivity allow you to detect down to 0.1% of the subclones. For the assay that we offer both catalog panels and custom panels that I will talk about in a second. Compatible with single-cell suspensions. If you're studying with solid tumor, it is also compatible with single nuclei input. At Mission Bio, we truly offer end-to-end solution from our instrument, the assays that we offer, including catalog panel and custom panels, as well as cartridges. And we also help you with data analysis. And our workflow is truly built seamlessly with the Illumina NGS workflow. Using our Tapestri platform, you can generate library that can be directly used on Illumina sequencers. And the FASTQ output file you get from the Illumina sequencers can directly be plugged into our software for data visualization and data analysis. And now let's just talk about the key technology inside of the instrument. As a microfluidic-based single-cell platform, Tapestri deploy a uniquely powerful two-step microfluid process to capture single-cell DNA content and reactions into single oil droplets. And the first step is cell encapsulation. This step captures cells and protease into single oil droplet, followed by protein digestion on the thermocycler. And this step is really important in terms that it helps you to gain access to the chromatin DNA sequence. And this step really sets us success for the downstream target PCR reaction. And the second step is barcoding, where we merge the cell lysates, the barcoding beads and reaction mix into another oil droplet through microfluid channels. And the reaction mix will be captured into a picoliter droplet for targeted PCR reaction. And to analyze protein, we simply incorporate a antibody staining step upstream of the whole workflow, which I will talk about in my last session. And in terms of the panel that we offer a wealth of catalog panels to get you started in both heme and solid tumor space. And we also offer custom design software, Tapestri Designer, that help you to design custom panels. If you're, for example, interested in studying rare genotypes or you're working in a gene editing field. And on average, our panels allows 100 to 300 amplicons in each panel, but we have worked with more than 1,000 amplicons for our custom panel. And here are just, again, some specs for our catalog panels to have you give you an idea of the panel that target different genes and how many amplicons included. One thing I really would like to point out is that, as you can see, that our catalog panel have really good panel uniformity. That means that this could significantly reduce your sequencing cost to recover those low performance amplicons. And our sequencing recommendation is 60 to 80x coverage per cell per amplicon. And actually, because we're using targeted method, it's not really a huge of sequencing cost that you will need. And we recommend, again, sequencing on Illumina platform with 2x150 base pair variant sequencing. And lastly, we offer a whole suite solutions for you to perform analysis as well, from automated panel design using Tapestri Designer, primary data analysis using Tapestri pipeline, all the way to secondary data visualization using Tapestri Insights. And lastly, I would really like to highlight some of our publications. Back in 2008 and 2018 and 2019, we had our first 5 publications that came out. And as you can see that our application expands in both heme and solid tumor space. And the study focused on therapeutic resistance, clonal remodeling after patients get bone marrow transplantation. And already in 2020, we have 5 new publications in Blood and Blood Advances journal. And we also have 5+ publication in the preprints as well. And the research topic also really expanded into MRD detection, biopharma drug studies, multi-omic studies, and also even gene editing research. Without any further ado, I'd like to dive into our custom user case. First, I would really like to share a very recent publication that highlights our collaboration with biopharma Agios. This study is to study therapeutic resistance for the drug that they have made, ivosdenib. So IDH1 mutations occur in 6 to 10% of the patients, and Agio makes this drug that's IDH1 inhibitor that has been approved by FDA back in 2018 to treat patients with relapsed refractory AML. And in the follow-up study that reported in a journal published in 2018, they identified patients that developed resistance to this drug. And we really like to look at the molecular mechanism that underlies the resistance and relapse in the subsets of the population that didn't really respond to ivosdenib monotreatment. So first, I really would like to show you a genetic landscape of those 174 patients that they have selected for this study. Note that this genetic landscape is using the data from the bulk sequencing. So as you can see here that the patients are listed from left to right based on their response to this drug from best to worst. And on the y-axis, you have all the different mutations listed from top to bottom, and they are categorized into different sections based on their functionality. And as you can see here that the mean number of the mutations tells you that the genomic complexity really is associated with the different incomes of the patient. And the more complex the genomic mutations is mostly associated with the worst outcome. And similar to other groups finding, the mutations mainly located on the RTK pathway. And lastly, also very importantly, Agios really would like to understand the cohort that acquired IDH2 mutations because it plays a redundant role to IDH1 in the oncogenesis pathway and really could account for the ivosdenib resistance and relapse. Moreover, Agios actually has a drug that serves as a IDH2 inhibitor and could be potentially used in those relapse instances. So as you can see here that they have identified patients that had both IDH1 and IDH2 mutations. However, whether those mutations occur in the same single cell level is not known by bulk sequencing. As such, they turn to Tapestri for help to better understand the clonal architecture heterogeneity at a higher resolution so that patient can understand better. And as you can see here that they picked 9 patients and 6 out of 9 patients have developed resistance after they acquire IDH2 mutations at different single cell. And some of the patients at the same single cell along with the IDH1 along with NPM1 and FLT3 mutation. And some of group of the patients acquired IDH2 but at different single cell level. And lastly, they also identified some patients even already had IDH2 mutation at baseline level, and they expanded at different cells and expanded during relapse. And second user case I would really like to highlight is a study that we collaborated with Dr. Ravi Majeti's group. And this paper is also recently came out of during 2020. The study was in collaboration with their group from Stanford. In this study, they would really like to explore the potential usage of Tapestri platform in MRD study. As we all know that the current popular method to detect MRD is FACS PCR, qPCR. However, Tapestri does have the advantage to elucidate clonal architecture. And moreover, it can distinguish different clonal relationships, for example, CHIP and MRD. I will talk about that later. And in this study, they selected 14 patients at 3 different time points. And the key question they would really like to answer here is, can the patterns of MRD and clonal evolution be detected by using Tapestri single-cell sequencing platform? So here I would like to show 3 different patient patterns. And the first, they were able to detect it using Tapestri single-cell DNA sequencing platform. And the first, as you can see, is a very typical disease relapse model that the patient holds the same pathogenic clone at baseline and relapse. And the second one, the patient had a clonal switch event where they had different clones at their diagnosis and relapse stage. And the third patient, very interestingly, they had a more complex situation that they had both CHIP and MRD. And our platform, Tapestri, was able to distinguish between those two and help better understand this patient's clonal diversity. And in the end, this is how he imagined using single-cell sequencing technology can potentially help with future MRD study. Basically, the patient after single-cell sequencing, this may allow clinicians, based on their clonal diversity, this allows clinicians to better stratify the patient based on their clonal diversity. For example, a patient with decreased clonal diversity at remission may be associated with longer relapse-free survival. And moreover, the detection of MRD would be very, very important for the clinician to make the call to escalate or de-escalate, and even in clinical trials. And lastly, because I don't really have time here, I just would like to point out that although I highlight heme publication here, Tapestri platform also has the capability of using single nuclei as input. And we do offer a universal nuclei extraction protocol that can be downloaded from our website. And we have tested with various tissue types, including frozen fresh samples and OCT embedded, and all of them have really good performance and cell output. Lastly, and most importantly, I would really like to talk about our recent advances in the multi-omic space. In 2019, single-cell multi-omic has been voted as the Nature Method of the Year, and that's really coincident with Mission Bio's roadmap as well. So at first, we launched a CNV analysis in mid-2019. Using our targeted approach to detect CNV using both read and genotype, we really gained the advantage of lowering the sequencing cost, but with a high resolution comparing the traditional single-cell genome amplification method. And moreover, using our platform, you could detect CNV and SNV at the same cell level. We did a cell mixing study to further apply our, to better understand our method. And as you can see here that we mixed 4 different cell type with different genetic background and using Tapestri and sequenced them using Tapestri platform. We were able to distinguish those 4 different cell lines just based on their copy number, gain and loss, even if Raji and Jerka had only singles CNV difference. And translated into a t-SNE plot, as you can see here, that the 4 different cell lines were able to be distinguished, and we colored them actually on this based on their SNV. And on the right-hand side, we are also able to detect the SNVs of the 4 different cell lines, and moreover, we can detect their zygosity status for different mutations as well. And we then launched protein analysis capability later 2019. And great news is that we collaborate with BioLegend on, we're going to collaborate with BioLegend on distributing antibody panels for protein detection. And right now we are the commercially only available single cell platform that uncovers SNVs, CNVs, and protein expression from the same single cell level. And currently, the BioLegend team, currently we distribute our antibody as well, but once BioLegend comes on board, we'll be able to offer panels that allow multiplex up to 40 plus antibodies for cell surface protein expression. And as we mentioned earlier, the whole workflow just has a small modification, including a simple antibody staining step up front of your whole workflow. And why do we want to do multi-omic study? This is just a graph demonstrating that this is especially true for AML research. As you can see here, AML is a cancer that evolves from uncontrolled proliferation from undifferentiated myeloid lineage cells that acquire sequential mutations. It involves different cell types and different mutation acquisitions. So using multi-omic studies, it really helps us to better understand how AML progresses. Dr. Ross Levine, one of our key opinion leaders in MSKCC, he definitely utilized this, our newest feature, doing multi-omics research to study AML patient. And he actually already have this paper in preprint available, and recently it got accepted. In this study, he profiled 123 patients using single cell mutation and protein analysis. And the whole study includes 7 protein targets, along with 109 DNA amplicons, which is a custom panel that we built for him. And in this study, he really would like to understand the order of the mutation acquisition in the AML patient and how these mutations correlate with different cell protein expression. And really, I would like to show you, highlight this multi-omic approach that he used to understand the AML patient progression. So here is an example that he was able to determine different clusters of the one of the patient sample based on their genotypes. And as you can see, after he overlaid the protein expression of CD11B, and the high expression level of CD11B in this cluster is strongly associated with genotype cluster with RAS mutations. Lastly, to summarize, using Tapestri platform with our first and only platform as the first and only platform to co-detect SNV, CNV, and protein at the same time in single cell, we are able to help the researchers to better understand both genotypes and phenotype at the same time. And the whole workflow only has a very simple cell staining protocol up front of the whole Tapestri workflow. And we have validated our multi-omics research both on cell lines as well as primary patient samples. And thank you for the webinar today. Feel free to let me know if you have any questions.
Hi, Yue. Thank you so much for a wonderful presentation.
Thank you, Kelly.
So we do have a few questions that are coming in, and also we love to hear questions, and we encourage you to use the chat or the Q&A box, and Yue will. So first question, does the platform work with mouse samples?
Great question. Yes. So we actually recently launched our Tapestri platform, Tapestri Designer 2.0, that help you even design custom panels based on mouse genome. And we also had a case study that you can download on our website, where we worked on mouse model in collaboration with Dr. John Lee from UCSF. So if you're interested in working with mouse model, the answer is absolutely yes.
Oh, great. Another question, I think it's technical. How many cells do you need per sample?
Yeah, great question. So it really, so for our hard threshold for the cell number that you need to put on our cartridge is at least 100,000. But if you are working with, for example, you're only interested in studying single cell DNA, then we recommend you start at least 500,000 samples, because our workflow does involve, cell washings. So you could probably lose some cells before you even load the cells on the cartridge. But if you're interested in doing, single cell DNA as well as protein studies, we do have a higher cell number recommendations, up to 2 million, but 1 million optimal. So because as we mentioned in this slide, that we have a cell staining protocol that does when you perform it, you do lose some of the cells during this process.
Great. Another technical question. How are the cells encapsulated?
Great question. So like I mentioned during our early slide, we have this novel two-step cell encapsulation protocol. I can reroute to my slide. So we do have this novel two-step cell encapsulation step. The first step is to, and first thing first is that we are a microfluidic based single cell platform. So we capture cells into oil droplets. We have this novel two-step. First step is to encapsulate cells into oil droplet, and second step to encapsulate barcoding mix and reagent mix along with cell lysis into another oil droplet.
Another technical question. What viability do you need your cells at? They know that it's important for RNA-seq. How does it correlate for DNA-seq?
Yeah, great question, Kelly. So as we talk about working with DNA, you do have the advantage working with a relatively stable genetic material. So we recommend 80% viability. However, we have worked with even less than 20% cell viability, and it has excellent performance, high cell throughput, high panel uniformity. So it really depends. Although, like I mentioned, we recommend 80%, but if you really have good cell integrity, although it has a low cell viability, we can still work with that.
Great. All right. Well, it looks like we don't have any more questions. Yue, thank you so much for giving a wonderful seminar. And if anyone has additional questions that they think of after the webinar, please feel free to email us. Hope everyone has a great day, and thank you so much, Yue.
Thank you, Kelly. Thank you, everyone. Have a good evening. Good day.
Bye.
Bye.